Now next type of chromatography is gel filtration chromatography, also called molecular sieve chromatography. Principle: in gel filtration chromatography, the separation of particles is based on their size and molecular weight. The apparatus consists of a column packed with sponge-like gel beads, usually cross-linked polymers or copolymers, generally of either styrene or divinyl benzene, containing a narrow range of pore sizes. The gels serve as molecular sieves for the separation of smaller and bigger molecules. The solution mixture containing molecules of different sizes is applied to the column and eluted with a buffer. The larger molecule cannot pass through the pores of gel beads and therefore move faster through the void space or interstitial space between the bead particles and will appear first in the eluate. As told earlier also, eluate is the fluid or mobile phase which is exiting the column. Now the smaller molecules enter the gel beads and are left behind as they pass down the column by entering one bead to another. Therefore, the smaller molecules come out slowly out of the column. As you can see in the diagram, when a sample is applied to the column, the high molecular weight compounds will flow down the column very fast by passing through the void volume or interstitial spaces present between the gel beads, whereas the smaller molecules will enter the gel beads because the gel beads are porous, and therefore the low molecular weight molecules will pass from one gel bead to another and taking more time to come out of the column. The size of the pores of the gel beads can be managed by taking different concentration of the material used for making this gel bed. Now let us see types of gel filtration chromatography based upon the types of molecules separated. First type is the group separations. In group separations, the component of a sample are separated in just two major groups according to the size range. This type of group separation can be used to remove high or low molecular weight contaminants. As you can see in the chromatogram, that when a sample is injected, the components of the sample mixture are separated into high and low molecular weight components. Gel filtration based on Cefadex enables group separation of biomolecules that are above the exclusion limit of the medium from contaminants such as salt, dyes and radioactive labels. The size is referred to as the exclusion limit, which means that molecules above a certain molecular weight will not fit into the gel pores. Molecules with sizes larger than the exclusion limit do not enter the pores and pass through the column relatively quickly by making their way between the beads, that is through the void volume or interstitial spaces. For example, it is used to separate phenol red from culture fluids or small molecules such as excess salt, also known as desalting, from the protein mixture. Cefadex G10, G25 and G50 are used for group separations. These different types of Cefadex vary in their degree of cross-linking and selectivity for specific molecular sizes. Next type is the high resolution fractionation of biomolecules. It is used to separate multiple components in a sample on the basis of differences in their molecular size. The goal is to isolate one or more components and to determine their molecular weight. The best result for high resolution fractionation will be achieved with samples that originally contain few components or with samples that have been partially purified by other chromatography technique in order to eliminate molecules that are of no interest. It is used to separate monomers from aggregates to determine the molecular weight or to perform a molecular weight distribution analysis. Polymers inherently have a distribution of chain lengths resulting in a molecular weight distribution which can be determined using this type. Now let us understand the working of gel filtration chromatography. Here, first spherical particles of gel filtration medium are packed onto the column. The sample is then applied to the column. Buffer or mobile phase and sample move through the column. Small molecules diffuse in and out of the pores of the matrix and so stay longer onto the column and are delayed in their passage down the column. As buffer passes continuously through the column, molecules that are larger than the pores of the matrix are unable to diffuse into the pores and pass through the column first. Separation occurs at different intervals which are followed by detection of components. Samples are eluted isocratically, that is, there is no need to use different buffers during the separation. However, a wash step using the running buffer is usually included at the end of the separation to facilitate the removal of any molecules that may have been retained on the column and to prepare the column for a new run. As you can see in the diagram, that when the sample mixture is applied onto the column, the yellow beads, being large in size, passes through the column first by passing through the interstitial spaces between the gel beads, whereas the small purple molecules enter into the gel beads and pass through the column by entering one bead to another, and therefore takes more time to pass out of the column. The fractions are collected at the bottom in test tubes at a fixed volume and at a fixed time. Now let us discuss the media selection, that is the type of media used for making the gel beads. Porous beads are selected based on their following properties, that is chemical and physical stability and inertness, that is lack of reactivity, and adsorptive properties, that is the media that we are using for making the gel beads should not react with the compound to be separated and should not adsorb the compound, because we want our components to pass within the gel beads. The following table shows the trade name of the different media along with the type of polymer they are, the types and fractionation range that they offer, and the uses. Cefadex is a polymer made of dextrin cross-linked with epichlorohydrin. It has many types, for example as G10, which separates biomolecules such as peptides of molecular weight more than 0.7 kilodaltons from small molecules of molecular weight less than 0.1 kilodalton. Cefadex G25 is recommended for majority of group separations involving globular proteins. It removes salt and other small contaminants away from the molecules that are greater than 5 kilodaltons. Cefadex G50 separates molecules of molecular weight more than 30 kilodaltons from the molecules which have a molecular weight of less than 1.5 kilodaltons, such as labeled proteins or DNA from unconjugated dyes. G100 separates molecules of more than 150 kilodaltons. G200 separates molecule of more than 600 kilodaltons. Cefacryl is the another media that is made by cross-linking dextrins, which is suitable for fast, high recovery separations at laboratory and industrial scale. Ceferose is made from agarose, which is suitable for the initial purification of ribonucleoproteins from cell crude extracts. Biogel is made from polyacrylamide, which are extremely hydrophilic and essentially free of charge and provide efficient, gentle gel filtration of sensitive compounds. Superdex is made of dextrin cross-linked with agarose, which is the first choice for high resolution, short run times and high recovery. Fractogel is made from polyvinyl chloride, which is suitable for fast, high recovery separations at laboratory and industrial scale. Superose is made from dextrin cross-linked with agarose to various extent, is capable of separating small molecules like peptides and single amino acids at laboratory scale. All these polymers have a different fractionation range and thus forming different types. Now let us see the applications of gel filtration chromatography. The first one is purification. The main application of exclusion chromatography is in the purification of biological macromolecules by facilitating their separation from larger and small molecules. Viruses, enzymes, proteins, hormones, antibodies, nucleic acids and polysaccharides have all been separated and purified by use of appropriate gels or glass granules. It is also used for relative molecular mass determination, which is done by using a calibrated column with substances of known molecular weights. It is also used for separation on the basis of size by selecting the gel beads of different porosity. Sugars, proteins, peptides, rubbers, etc. can be separated. It is also used for solution concentration. Solutions of high molecular weight substances can be concentrated by the addition of dry Cefadex G25. The swelling gel absorbs water and low molecular weight substances, whereas the high molecular weight substances remain in solution. Another application is desalting by use of a column of, for example, Cefadex G25. Solutions of high molecular weight compounds may be desalted, that is removed from contaminants such as salts, detergents, lipids, and chiotropic agents. The high molecular weight compounds move with the void volume, whereas the low molecular weight compounds are distributed between the mobile and stationary phases and hence move slowly. This method of desalting is faster and more efficient than dialysis. Applications include removal of phenol from nucleic acid preparations, ammonium sulfate from protein preparations, and salt from samples eluted from ion exchange chromatography columns. Gel filtration can also be used to facilitate the refolding of denatured proteins by careful control of changing buffer conditions. It is also used in protein fractionation experiments, and it is also used to determine the quaternary structure of purified proteins. Now let us see the advantages and limitations of gel filtration chromatography. Advantages include that gel filtration is a robust technique that is well suited to handling biomolecules that are sensitive to changes in pH, the concentration of metal ions or cofactors, and harsh environmental conditions. A significant advantage of gel filtration is that conditions can be varied to suit the type of sample or the requirements for further purification, analysis, or storage without altering the separation. Separations can be performed in the presence of essential ions or cofactors, detergents, urea, guanidine hydrochloride at high or low ionic strength at 37 degrees Celsius or in the cold room according to the requirements of the experiment. Unlike ion exchange or affinity chromatography, molecules do not bind to chromatography medium, so buffer composition does not directly affect resolution, that is the degree of separation between peaks. It has a short analysis time, it gives a well-defined separation, it shows narrow bands and good sensitivity, there is no loss of sample, small amount of mobile phase is required, and the flow rate can be set. Limitations includes that only limited number of peaks can be resolved within the short time scale of the run. Filtrations must be performed before using the instrument to prevent dust and other particulates from running the columns and interfere with the detectors. The molecular masses of most of the chains will be too close for the separation to show anything more than broad peaks. That is, if the molecules which are to be separated have similar molecular weights, the chromatogram will show broad peaks.